gone into the test kit business, which also surprised me somewhat because I think the last time we were talking, you were more of a electronic hardwares guy,
Mm-hmm.
and I want to ask you about how you got into the test kit business and also how you assess the current situation. If I can have that for my, probably do a Twitter strand on it and just for my background. Like,
Yeah.
how do you think Taiwan is coping with the current crisis? Whether it's appropriate that we are remaining at level 3, which
Yeah.
is a soft lockdown, and if you can also sorry, I know I'm hurling too much question at you at once. I'll
So
stop in just 1 minute. But also, I think I'm particularly curious about how you think the government has handled the backlog from testing, which is to add it retroactively to the past week.
wow, that's a lot. It's a lot to unpack there.
Yeah, I
Yeah.
know. Please feel free to take it one at a time
Yeah.
and just start wherever you feel
Right.
comfortable. And we have time to keep going on. And if there anything that's like not in your wheelhouse, just tell me and we can move
Right.
on.
Okay. Yeah. So I just need to clarify that I'm not technically right now. I'm not in the consumables business for test kits. So yeah, so you're right. Basically, I was working I work in electronics and design more. So
Mm-hmm.
yeah, so this is really I started working in a vaccine company here in Taiwan. So I did my 5 years in AMI, which is the American company, right? So then I got my
I'm sorry, you're gonna have to. I haven't. I'm not aware of that company.
yeah. So AMI, it's a BIOS company, a BIOS and motherboard company. So we do firmware and software and hardware for all of the Taiwanese motherboard companies, basically.
Okay,
Yeah.
good.
So when you
Uh huh.
boot up your computer, you'll see AMI. So I was
So how does
running that.
that connect with vaccines?
Yeah. So this company, I was transferred. I took a transfer to Taiwan as their vice president for 5 years. And at 5 years and 1 day, I filed for my uh, permanent resident APRC. So when I got that, I was free to work for any company, right? Yeah, so I did my 20 years in AMI. So after that, I actually always wanted to work in biology. So I did a startup in a vaccine company called Medigen.
Medigen.
Yeah,
Wow. Uh huh. Mm-hmm.
yeah, or as technically it's SBC, Schwarzer Biotech. So I was working there for 3 years.
When did you start working for Medigen?
This 2000. I'll have to check exactly if it's important. It's yeah, I can give you all those dates. Is in
Yeah,
my
no worries. It actually doesn't matter. I was just
yeah
curious to try and get a picture of where you were at.
yeah. We'll see. I was in 2015. May
2015.
Yeah, May 2015.
So you always wanted to work in biotech. So you made the switch
Yeah,
to vaccines. How did you do that? Did you already have any background, or did you make a lateral move based on your expertise in any transferable expertise from hardware? How did that happen?
yeah. My education is really physics and chemistry and biochemistry. That's my background. So I kind of started in physics and chemistry, but then I ended up in electronics. So now it's kind of
Got
going
it.
back to this, what I always wanted to do. But so I had this combination of electronics and biotech, basically. Or so so for them, I was working on PCR. So I was designing PCR machines. So they 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 obviously make vaccines, but they also make reagents for PCR. PCR is something that everybody knows now, right? In the past year.
Yeah, I'm
Yeah,
sorry, but you're still gonna have to unpack it for
yeah,
me. I know it's a kind of testing, rapid testing.
right.
What does PCR stand for?
Yeah, so PCR is a polymerase chain reaction. So this is a
Polymerized chain reactions.
yeah. So these
They're
are
the fast tests, right? Or are
no,
they the slow
those are
tests?
those tests. They take hours for the actual process. Yeah, the actual machines, they usually, traditionally at least, they take hours to do the testing because they have to amplify the DNA in the sample if it exists. Like if there's if the target DNA is in the sample, they will cycle it over and over again to amplify it. So the way it works is if the pathogen you're looking for in a PCR sample is in the sample, then it will be amplified. So every time you run a cycle, it gets amplified.
Are you still working for Medigen right now?
No. It's been no, not anymore. No. So I did about 3 years there with working for doing PCR and PCR machine design and some other projects. Actually working on patents for tracking growth in farm animals, like AI. Using cameras to observe animals, how they grow over time. So working on algorithms for that, and for fish, like using cameras to track the growth of fish on the water. They did a lot of technology projects, but in biology, right, in the setting of biology. So yeah, so I was working on PCR, and then we tried this. We tried to do PCR. We tried to take the PCR machine, right, out of the lab and into the field. So the idea was we would design a PCR machine that we could actually send to the farm. The fish farm, the pig farm.
To utilize an early warning system for any kind of
exactly.
disease.
So it's design a PCR machine with a computer, with a GPS, so that the farmer or people on the farm. So the normal process is that you take a swab, you get a veterinarian to come. If you have a sick animal, you get a veterinarian to come to the farm, then you do a swab of the animal, take a blood test, do a biopsy, whatever you need, right, depending on what the illness is. And then you put it into a viral transport medium, send it off to a lab or university. And that could take weeks, Traditionally it could take weeks, but then maybe a week, right? But I think, sometimes you can get it down to days, but it still takes a long time. So the idea was we take the machine, we design a machine that you can put into the field, and then you can make it simple enough that the farmer or whoever cares that, or at least somebody in the farm could do this sample collection and put it into the machine, and then right away you can get a reading for the disease in the farm, and it would be geolocation based, and it would give you a pin on a map, like what kind of pathogen was tested exactly where,
And it would be within hours?
Within hours, yeah.
Within hours.
Exactly,
Great.
yeah.
But it would also be located within the field, so it
Right.
could be the sort of thing that they run every day, so they can get pretty much, as soon as an outbreak occurs, they can do something about it within the day.
Yeah, exactly. So this is where when you have a sick animal on a farm, right? For example, I grew up in Norway, right? So there's a lot of salmon farms. Right? So typically the way it works, you have one dead fish floating up to the top. So you take that one out and you try to figure out what happened to that fish, and then by the time you get it out, there's 10 more dead fish, right?
Yep.
And then there's the rest of them, because this can happen very quickly. So time is of essence here. So we got this PCR technology down to doing a complete series of cycles in less than an hour.
Wow.
So like down to 40 minutes or so. I mean, that was back 5 years, more than 5 years ago. So we got down to
aquaculture is, well, I'm sure you can export it all over the world, but aquaculture is a big deal in Taiwan. So
right.
Definitely a present market.
Yeah. So what happened then is that you take this machine. The idea was to take this machine, PCR machine. You make it portable. You put it, you put a GPS in it, and have a cloud backend system where you can track where all these machines are. So I even made it. I even outfitted it with something called an indoor GPS.
What is an indoor GPS?
It's something that typically if you're using your GPS and you walk inside, you'll get a voice will say, GPS signal lost. Because you don't have satellite inside, right? So I made a special antenna that it's very big antenna that folds up inside the machine, so you have a higher chance of getting satellite reception even inside a building. So you could use it in a not necessarily in the basement of a tall building, but you could use it inside a farmhouse, for example, where you don't have
Got it.
direct
Okay,
line to the
so
sky.
for instance, if this farmer only had one of such a machine and he has 4 fish ponds,
Yeah,
he can bring it from pond to pond and the geolocation will still work even if the fish ponds are covered.
right. Where the machine
Got
is
it.
covered. Yeah, so it's just just a way to get more accurate reading for where the machine were.
So this sounds all very well and good. You guys are
Yeah,
on your way to conquering the fish PCR
yeah, and
aquaculture market.
yeah, and pigs too, and
And
goats
pigs and chickens,
and
yes.
chickens.
You
So
know,
we're gonna
it's really
sell these.
amazing the degree of automation Animal Farm is. The 70% of the cost of a modern chicken farm is feed.
Yeah,
Very low labor,
yeah, I believe that.
very automated.
Hmm.
So I'm sure any kind of machines like that would aid in keeping the animals healthy. Because basically they just are decreasing the amount of labor input as much as possible.
Yeah.
But how did you guys go from there to PCR test for humans?
Yeah, so it's funny that the machines doesn't actually care if you test on human or animals.
Okay.
It's you can probably use the machine to differentiate if you have a human or an animal, by genetic by
Hmm.
limiting the genetic area that it's looking for, right? But you know what happened is it just didn't work.
What didn't work?
It just didn't work. You couldn't take the PCR. It was so hard to take the PCR machine out of the lab and put it into the field. You just couldn't do it. It didn't
Oh,
work. It was a failure.
oh dear.
Yes.
That's terrible. Even with
Well,
indoor GPS and everything. How come? Why? I don't
well,
understand.
well, that's the thing. This is PCR technology, right? So PCR is extremely sensitive. PCR is really great when you really need to look for something very specific, like a very specific sequence, and you have a very clean environment. So the instructions for using such equipment is that you have to have a very clean lab. You have to wash it down every time. All your reagents and your canisters, everything you use, has to be completely sterile, right?
And
So
that was when the field
yeah.
conditions didn't match the lab conditions. Okay,
Every
so
every test you do for any organism becomes positive if you do it outside in a lab, especially in a farm, because it's just too much.
all right, let's
Yeah.
gloss over this then. Our time is
No,
short.
it's
Let's
great.
talk about
It's a
how
great experience, right?
it
Yeah.
was a great
Yeah.
experience. And
Learned
now
a
let's
lot
talk
about
about
PCR.
let's
Yeah.
let's 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 rewind up to the present moment. What are you doing
Yeah.
right now?
Yeah. So then, I was actually working on graphics, graphic cards, GPUs, and after that, I started a company to do graphics cards, basically. And graphics cards is also very powerful. Not using graphics, using mining or
Yeah,
yeah,
isn't
AI.
graphic cards a thing that's been used a lot for Ethereum mining?
Sure.
Mm-hmm.
Right. So, I mean, I didn't really design the graphics card. So again, I was designing something that ended up being used for something else. So we sold a bunch of cards for mining, but that wasn't the purpose. It was really the purpose to design these cards for very large video screens, like outdoor signage and things like that. So most of the
Mm-hmm.
cards we've been selling was to big companies, parks, like museums, huge installations like museums and outdoor signage, this kind of stuff. So it's a very high end digital signage stuff. But the
Okay.
cards ended up being used for also for AI, artificial intelligence, video processing, deep learning, things like that, because of the GPU aspect of it, right?
That's cool.
Yeah. So then when I was actually went to a trade show in Europe when the whole pandemic started in Wuhan.
Oh, okay.
So
Uh-huh.
I was actually booked. It started in December, right? Uh,
Yeah.
kind of when we first knew about it, and in February I was in Amsterdam. I was still doing graphics, and then of course we heard about it. I made it just I just made it back to Taiwan before they started locking borders and things like that. So, and then I had some friends in Atlanta. I used to live in Atlanta, right? So
Oh, I didn't know that.
yeah.
Uh-huh.
So I lived in Atlanta before I came to Taiwan. So in Atlanta, there's a lot of biotech, and I used to live not directly near the CDC, but in the area there's a lot of people who work for the CDC in biotech. So I had a lot of friends in that business. So I got in touch with somebody got in touch with me to see if we can design a machine because they know I used to I worked in biotech.
Yeah.
And so they actually wanted us to make a machine to read lateral flow essays. And lateral
To read, so sorry.
lateral flow essays, right? So the original one was this one here. This is a lateral flow essay.
Mm-hmm.
So you take a sample from somebody and you just put it into this window here.
Yeah.
And then traditionally this is almost like a pregnancy test that you just have to you can just read the lines here, right?
Yeah. Yep.
But these things ideally needs to be read by a machine, not by a human eye, because there's a lot of mistakes. If you read 10,000 of these every day, or even hundreds, your eyes gets tired, you make mistakes, and we've seen that. It's very easy to make mistakes, especially when the viral load is low. So I actually started designing a machine this is from a company called BD Veritor, so they already have a machine, but their machine can only read this one. And there's another company who make a machine who can only read that one.
Got it.
Yeah, and the machines that they have are not designed for high throughput. They're designed for one person comes to the doctor. They sit down to do. The doctor thinks maybe they have flu, maybe they don't. So let's do a quick lateral flow assay and see. And then they take the sample, wait 15 minutes. And you get the result, right? So they're not designed for doing onesies and twosies at a time. In one day they may use the machine once or twice. Now we're in the situation we need to test, thousands of people in line, as they come through the airport, as they go into the factory, as they go into school. So the need became there for a machine that can read these test kits very quickly. These takes like 10 seconds to read.
Can you hold up one of those?
Yeah, sure.
Kits, please.
Yeah.
Just a little bit further away from the screen. Further away.
Well, I can send you a better picture if you want.
No. Yeah. I just want a picture of you holding it. I'm doing a screen cap for LinkedIn.
Okay.
Have an image. Okay, that's good.
Right. There's probably other. I can. Let's do a better. Yeah, I can work on something.
Yeah, sure.
Yeah. Let's do a better.
Don't get too hung up on it. I just want to because all my LinkedIn posts do better if they have an image.
Oh, I totally agree. Yes.
That's why I wanted to have one, but it's not a big deal at all. So
a pure LinkedIn post with just doesn't do well.
It doesn't do well.
If you share somebody else's, somebody else's picture article, it does okay. But if you have your own personal image, it does by far the best.
The best. Yeah. That's what I find, and so I wanted a image. But let's get back to the interview though. So you're basically doing a machine that will read the test, and you want your machine to be universal and to have high throughput. Because right now, what they're doing right now, are they using the machines for each proprietary brand, or are they using humans?
Well, this is the first one actually. This here is a better screen capture here.
Okay, here we go. Let's take a look. Hold it up a little bit higher. Okay. Great. That's good. That's a better. So
that's the actual machine. Yeah.
That's the actual machine.
Yeah.
So you would.
Yeah. So you just put it in here, and that's it. And then you put the next cassette, which can be a totally different cassette. So
it could take cassettes from different companies.
Yeah. Or it can take. This is like these are the. when I started, these are the two most popular ones. This is from the U.S. So this is BD Veritor and this is Quidel. So if you use their machine, you can see that these are quite different, right?
Yes.
They're a whole different geometry. So I actually designed something that's very high throughput and very durable. So this one fits. This one fits. So all of these different lateral flow assays fits.
And what is the throughput rate for a machine like that?
Couple of seconds per read. It reads.
Oh, really?
Yeah.
Got it. Right now, how are the Taiwanese authorities reading their tests?
They're not using any machine yet.
They're not using any machine yet.
Well,
how quickly can you get your machine online for them, and how would that improve the throughput rate? Huh?
Next week.
Next week. All right. That's very, very exciting.
Yeah, it took so long to get FDA approval, so we're hoping. We've been pushing. The government's been helping us now because this has to be approved by the FDA. And because we're not using an emergency use authorization like an EUA. So with the EUA, that's an emergency use. You don't need to have a factory authorization then, like what's called GMP. So, but when we filed the EUA back in December at the Taiwan FDA, then they recommended us that we should go for a Class 1 medical device, not emergency use authorization. And so we did. But that means we had to go through a lot more regulatory approvals, of environmental, RFI, regulatory testing.
Well, it sounds like you guys are coming online in time to make a difference, though. Can you talk
just in time?
Can you talk a little bit about how this could help Taiwan contain this current coronavirus outbreak, and whether you think level 3 is enough? I mean, I know you're not the expert in that. It's your personal opinion.
Not really qualified to answer that, but I had a lot of experience over the past year, when it comes to on-ground testing, right? So obviously with PCR, you'd have to send your entire workforce to a hospital because, again, my experience from PCR, right? You cannot do it. You cannot send these PCR machines out to the field. That was the big takeaway from, But this machine, because I learned that, right? And I figured out how to do it. So this machine you can put anywhere. You can put this in an airport, in a school. You can put it in a farm. So one big thing about this testing is we already have companies in Taiwan who are building lateral flow assays. Typically the local companies. These are like the fancy, expensive ones. We
already have a local test solution for the little sticks, right?
Yeah. This is a local company. Much cheaper, just as good, works the same. And, they have these now for African swine fever and chicken flu or bird flu. Yeah,
so right now are the, first of all, what are the technical name for those sticks?
Lateral, they're called lateral flow assays.
Lateral flow assays, and they're using PCR.
They're not using PCR.
They're not using PCR. Lateral flow assays. Is that a different technology? And
totally different.
Totally different, and they're super quick, right?
Yeah. So PCR actually looks for a particular piece of DNA or RNA. Right? It's a molecular. It compares a piece of molecule to another piece of molecule, and if they match.
How does lateral flow assays work?
There's actually a strip of paper. I can just open one up so you can get a feel for it. So this is a test cassette. So the chemical reaction actually happens in the field on the strip, unlike a PCR, which is happens in the lab in a big machine.
Yep.
But if you take one and open it up, there's a piece of paper in here. So I'm going to take that piece of paper out too. So here's a piece of paper. So this is the analyte pad right here. So you basically, the other way around. So right here you put some drops. So there's a hole in the here.
Yep.
Right here. So you put some drops of liquid in there. The liquid has a buffer in it. The buffer does a couple of things. Like, so you take a swab from somebody's nose or a gum line, depending on what, HIV is from the gum line. COVID happens to be from the nose, right?
Yeah,
So it just
it's pretty
depends.
intense. I've seen the videos. It's like
Yeah.
they have to really get in there.
Right, but we fixed that. You don't have to do that anymore.
Oh, really?
Yeah, I'll show you.
Okay, please
Yeah,
do.
so
Because I was like, oh, that's not going to be fun when the time comes.
so when I started out, if I was just following, that's when you enter a new industry, you really have a chance to make some innovation. So I'll show you some of the innovation things. But basically here is that you have a few drops that you put, right? And that drop, I can actually give you the whole process. But this is a little bit important actually. Oh, I do these video calls quite a bit, so I usually have all the props here within the arm's reach. It's been a lot of video calls. So what you're referring to is this one here, which is a nasopharyngeal swab, right?
Yes.
So I'll compare this one to the size of my head, right? So yeah, so you can see, it's not very interesting. It's not very exciting. It's not something you look forward to doing, right?
No,
right.
Not in particular.
And everybody knows how uncomfortable that is.
Yes.
So imagine having to do this every day before you go to work.
Oh, well, that
every day.
Sounds terrible.
In order to be let into your university, you'd have to do this every single day. People just wouldn't come back to school if you had to do that. They just wouldn't show up for work, right?
Yeah.
So what we did is I work with some, that's what I love about Taiwan. There's so many suppliers. Anything you ask, somebody can make it, right? So I designed this one.
What?
Yeah. So
how does that one work? It seems like just like a lot shorter.
Right. And you see the little red mark there?
Yes.
That's where it goes up your nose. It goes up your mouth only this much.
Wait a minute though. So how do you get an accurate reading with something that only goes up your nose that much?
Right. Because when this, and this is just so, this is pharmaceutical and biotech. They've been doing it the same way for 20 years or longer, right? So nobody questions the method. So this is what everybody was doing because that's what you had to do for the influenza typically, right? Because influenza, would typically be, in the back of your throat, right? That's where the but COVID actually replicates in any kind of cells that have ACE2 receptors. So ACE2 receptors are these particular type of cells that regulate blood flow. So those type of cells, they, where do you find them in the body? It turns out you find them on the tongue. You find them inside your nasal cavity. So it's not about going deep. It's about finding where. It's
the
about
ACE2
getting to
receptors.
So finding where are the cells with ACE2 receptors. It turns out there's some right inside your nose.
So how has an uptake of the short swabs been in Taiwan?
Well, the thing is, like, first you have to do it, then you have to have clinical trials to convince that you have to compare. So I spent a good part of last year at least from August, September, October, November, at least 3 last month, November, December, October, November, December last year to do clinical trials in the US. We did a lot of clinical trials. We tested different lateral flow assays, we did lateral flow assay testing. We tested hundreds of swabs, compared against each other. We tested, what kind of material is better to absorb the virus, because you're actually not picking up the virus itself. You're picking up cells. You're picking up epithelial cells, like the outer layer of cells.
Yes.
Yeah. So those are the ones that you actually need to scrape them off. You need to get some fresh cells from the top layer, and then you need to get those cells to attach. So this material here is a bit porous. you know, or this material here is it's got that right porousness where it's a little abrasive and takes the cells off, and then when you put them into the liquid, which is the next part of it, these are buffers. So, so you go from the swab, you collect the epithelial cells, and you put into this buffer, and inside the buffer there's a solution. So that buffer does many things.
So it goes to the buffer solution that
Yep.
washes off the epithelial cells from the swabs.
Yeah, and then there's a lysing process that actually opens up the cell. It opens up the cell membranes and lets the goo of the cells out. And inside those cells, there could be virus particles.
Okay.
Right, so that's how you get to it.
And good. The part that I'm interested in is a class, I believe you. These short swabs are capable of testing COVID without, getting your nose poked by one of those big guys. How are they doing in clinical trials, and when will they become the industry standard?
Yeah, we're pushing very hard. We're working. It's. You think if you have a great idea and you tell people about it, everybody would just come running and want to do it, right? But it's a big convincing game. You have to convince the FDA, the CDC, and then you know what else? You have huge pharma who's been selling these for 20 years. They don't want you to come and sell these because they're still selling these for $1 a piece. So if you make this one for 1 NT. They don't want you there.
We definitely need to get some more attention on that and see if that can move the pipeline, because I think a lot of people are suffering for nothing.
Right.
In fact, all you need is a short swab up the nose. That sounds much more pleasant. But in any case, let's focus on the key takeaway here. The key takeaway is that for the there's a variety of made in Taiwan lateral assays. Sorry, lateral what assays?
Lateral flow assays.
Lateral flow assay tests that's being used in the quick test stations that are popping up all over Taiwan right now.
There are,
They're currently still reading them one by one, and that is probably causing a higher mistake rate, and it's also reducing the throughput. You have designed a machine that will speed up the reading process and also decrease the amount of error. And it's gone through the certifying process and probably will be put into use in the next week or so. All over Taiwan or in certain places?
Yeah, I mean, so nobody was interested in this machine until a month ago, right? Because there wasn't any code in. So here's the thing. First you have to convince, and you have to go certification, and it's expensive, and it takes time. You have to convince a lot of people. The process is slow, right? And there's so much stuff you have to go through, right? And I understand, because you want to make sure Taiwan is doing the right thing. I mean, you want to make sure there's a quality product that goes out the door.
Of course.
Absolutely agree with this.
Absolutely.
Yeah. So, but we didn't plan to do this in Taiwan because nobody. We've been fighting the whole game in Taiwan about PCR versus lateral flow assays, right? They've fought this battle in every country, right? Because you have the PCR camp, who is, it's a $5 billion industry, I think. It's huge, right? The test industry. So you're fighting against these cheap, rapid, convenient test kits, right? And there's lots of professors and people who have been outspoken about the advantage and stuff. So you have to fight the establishment, basically. So we had to fight. We had to do the same fight as they did in America and in Europe, to try to convince the authorities that they can use this new type, right? And here, suddenly, a few months ago, the cases started exploding. So the convincing has been a lot quicker.
Because they need the volume in test kits.
Right? Suddenly they need it, right? So now, now we have to quickly ramp up all this machine production. And what we had planned to do was to, these machines were really designed for export, right? But now it looks like we're going to be using a lot of them locally.
At home.
Yeah, at home, which is great, So,
How fast can you ramp them up? When will they actually see service in the field? This is what I
well,
need to know.
the good news is we've already manufactured our first batches, and they're sitting on the dock right now, just waiting for FDA approval.
Okay, and
Yeah,
so
yeah,
you told me earlier next week, right?
yeah. I mean, the machines are sitting on the in the at the factory right now, just waiting to be used. You know, just waiting for a piece of paper that's going
Okay,
from the E
okay.
tree
How many
to FDA.
machines are sitting in the docks right now?
I think we can make a couple of hundred machines. You know, um, right now I think we have about 30 machines.
30 machines
Yeah,
you can.
yeah. That
Okay,
we
so
have
you have
that.
30 machines that are completely ready to go.
Yeah,
You can make a couple of hundred machines as
yeah.
very,
Next
very
month.
rapidly. We are looking at them as soon as that piece of paper comes down, they
Yeah,
are ready to hit the field
right.
and make a difference. Well, what a great story, Claus. I
Yeah,
think this is really fascinating, and I think it's so amazing that you've taken your basic training in science and applied it to so many fields. You truly have had a fantastic career,
right.
and I really think also you've been so great for Taiwan because, like you said, we are full of wonderful smaller suppliers who will do anything you want them to, but sometimes they need a little bit of direction, a little bit of creativity and inspiration. And I think people like you truly um, help Taiwan be able to, you know, people like you are what are the people Taiwan needs to take us to the next level, right? We don't always have to have people in America telling us what to do. We can have local companies, local startups.
Yeah,
So I'm really glad we reconnected.
yeah. Thank
I'm
you.
gonna post a short LinkedIn post because what we discussed is highly technical.
Yeah,
As soon as I post it, I'm going to send a link to you. You can tell me if I got anything wrong so I can amend
hmm,
it quickly. And after all this is over, let's have a beer. It'll be
good
really great
idea.
to catch up. Yeah.
But you just remember, right? I'm just gonna give you a call. I think if you have time, if you have 5 more minutes, I just want to clarify a couple of things, right?
Sure, 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 sure. Please
Yeah.
do.
So, remember like right now they're testing 500 people per day, roughly, per day in one of these testing centers because they're doing,
Yeah.
it's really, really backlogged. This machine can test 700 people per hour. So if you're
Wow.
talking about magnitude difference here, plus you
Okay.
don't have the inaccuracy. So we did clinical trials in the U.S. where we compared. It's actually called preclinical trials, where you actually have to send kits to the. It's
Mhm.
part of the FDA approval process. Is we have to send these kits to trained medical professionals
Mhm.
to
Mhm.
learn how to do the clinical trial before you actually do the clinical trial. And we found that even trained medical professional, only 25% of them get it right when the test is weak positive. And that's the most critical part. Is when somebody is presenting very early on an infection, right? The viral
That's when
load
they're
is
the most.
that's the most
Okay.
one, because if you test today and your viral load is really, really low, right? But it's there. Tomorrow it's going to be really high.
Got
So
it.
it's so important to get those weak positives correct and to avoid false positives. So we've solved that by doing machine reading, right? So you don't have the problem with the eye fatigue and mistakes. That was one thing. I don't know if you had about PCR versus lateral flow assays.
Mm-hmm.
You know,
Please give me a short explanation. My
right?
understanding is that the PCRs are better and the lateral flow
Yeah.
assays are quicker.
It's
But
two completely different technologies, totally
okay,
different. It's not even in the same ballpark almost, I would say. So with
okay,
PCR, you're actually taking a component. It's called a primer. You actually take a primer, that is, you actually take the sequence of the virus that you're looking for, and or the opposite of it. Double helix DNA, right? So you're looking for one side, and RNA is just one side of the helix. So you're taking a primer, and then you have a viral component, which may or may not be there, right? So you put some sample, that's a swab from somebody's nose, into this mixture, and then you have some enzymes there that help the reaction go along. So you basically you break the cycle by heating up the temperature, and then the double helix would separate because of covalent bonds. They break at 95 degrees C. And then if the sample is there, that sample will actually attach and make a copy. So now you have two copies, and then you cool it down, heat it up, cool it down, heat it up. So every time you do that, you make a copy of the virus if it's there. So that's
It's almost like you're growing it.
Yeah, it's you're growing it. You're actually growing it
Culturing
inside
it.
a little tube, you know. And when you keep growing it enough, then it changes the opacity, like the liquid. You know, it's clear in the beginning, and then if the virus is there, and you keep cycling, the clarity of the liquid is changing because there's so many copies of the pathogen that you found in there. So when the opacity changes, you use a light source and a light detector on both sides of the sample, and that's how you can tell if it's a positive. That's PCR. You know, that's what I was designing in another company. But with lateral flow assay, you actually have a strip paper.
Yep, you showed me the strip earlier.
Yeah. So you actually put that on here on the paper. So this is just like any paper, right? When you wet the paper, the liquid would just wick up the paper. That's what we call a lateral flow. Right. So it flows in one direction. So it will flow. You know, the second part here is actually a filter, a little filter. So it's a fiberglass filter. So if there's things like hair or snot or like particulate matter in the sample, it filters it away. So when the liquid actually comes on the real nitrocellulose membrane paper, which is here, that paper is printed with antibodies. You know, antibodies, right? So you have antigens, or you have antigens and antibodies. So the paper is printed with antibodies together with some particles that, when they're attached to the antibodies, have one color or they don't have a color. But if they actually attach antigen attachment, they release these particles that change its color. That's how you can see the line. So the mechanism actually antigen antibody.
Okay, so it's a very, very different way of detecting whether or not the virus is present.
Yeah.
And so the PCR tests are slower. You need to go through all these cycles to culture the virus up to when it affects the opacity of the liquid. But with lateral flow assays, you have these antibodies that are locked in the paper. And if the antigen is present in the sample, it will trigger a reaction where the color will change.
Yeah.
And that's where it's like the pregnancy test. You see the line, but the weakness of doing the lateral flow assays in assessing them by eye is that the human eye gets fatigued, and sometimes, especially in the presence of low levels of virus, you could have false negatives because they get thrown away. And what about the false positives I've been hearing about? How do they happen in lateral flow assays?
Yeah, they happen typically for a couple of reasons. Ethnicities is in our experience, in our experience, or at least my experience, the issue is with ethnicities.
Say more.
People are not all exactly the same. You know, when it comes to the kind of secretion we produce in our oral cavities. Right? So it turns out, and we found out this by just testing people. So, you know, for example, my nose compared to some of the other people's nose in the lab have a different result.
But if I understand the science correctly that you described for the lateral flow assays, they are detecting for a specific antigen.
Right.
So how does your ethnicity contribute to the presence or, the mistaken presence of an antigen if you do not have a virus?
Right. So when you swab somebody's nose, right? The epithelial cells is just one part of the swab. I think that PCR have that same problem, right? So with PCR or any kind of biological testing, when you take a sample, if you take a DNA sample, right? If you scrape the inside of your mouth and send that to 23andMe or any of the services like Circle DNA or somebody, right? Then most of that sample is not you, right? Most of that sample is not your DNA. It's stuff you had for breakfast. It's
Okay.
it's bacteria in your mouth. The bacteria have, there's parasites, there's bacteria, there's viruses. Every bacteria has macrophages and viruses. There's a ton of stuff from all kinds of evolutionary biology is on that sample, right? Including your own secretions, whether it's saliva or what do you call it? Enzymes in the mouth, for digestive enzymes, right? All that stuff can be present.
Okay, so if I
yeah.
can, if I understand you correctly,
Yeah.
when you have a false positive scenario,
Yeah.
it's because one of the contaminants on your swab is similar enough to the antigen that they trigger the antibody on the strip to have a reaction.
No, actually, so this buffer here, this lysine buffer, also contains a detergent. So when you take a sample and put it into here, then it contains there's a lot of biological sample there, right? Like I said, it's your digestive enzymes, and it's some cells and stuff you had for breakfast, and all kinds of stuff is in there, right? In that sample. But so there's chemistry in here is a detergent that's supposed to take away those things, breaks open the cells. And the nasal
what
swab.
you're still not what you're still not answering my question is, how does a false positive happen? Because you have an antibody that is supposed to detect the antigen. What causes a false positive?
That's not the false positive part. The false positive part in this case is caused by all the other stuff in the sample. That's called cross-reactivity testing. So when you develop these assays, you have to make sure that the assays, they don't react to other things, other pathogens. For example, mouthwash, right? For example, smoking, nicotine, coffee, oral antibiotics, mouth spray.
What is the mechanism by which the cross-contamination happens? I'm sorry, the
false positive
reaction.
Right.
False positive, yeah.
Right. It's something in the person's saliva or secretion in the nose that's very thick.
Okay.
So what you do is you put a detergent into the buffer, because that detergent actually dissolves the human produced secretions, and when it dissolves it, you can actually get the cells out. And then when you get the cells out, then you use a lysine buffer to open those cells up. So the amount of, like, if you're in the lab, right, and you need to put enough detergent, you need to put enough detergent in the sample so that you can make sure that those secretions are dissolved.
Okay.
And then how much detergent you put. If you put too much detergent, that affects your sensitivity. So you don't want to put too much detergent, but you need to put enough detergent so that you can get all of that. So we did in Brass Monkey. I found that was the perfect place for this. So first we took, there was a lot of foreigners in our office.
Yeah.
From South America and Europe and all over the place, basically, right? When we tested all those people, this is early on when we did clinical trial, kind of preclinical trials. We tested a lot of different people. We found there's differences, because it's so easy to think that people are identical, but biologically there's differences. So when we actually tested people from Europe, like myself, I was producing false positive, what looks like false positive lines. It looked like I was positive, very, very weak positive in the beginning. So then what they had to do then, they had to adjust the chemistry of the buffer to make sure that the detergent component could dissolve all the
But
human.
that has
Yeah.
really has nothing to do with your machine. That has to do
Yeah.
with the lateral flow assay.
It has to do with the buffer, the detergent in the buffer, right?
detergent.
So
Okay.
particularly what happened in China early on is that they developed tests that they sent to Europe, and in China the tests they worked perfectly, right? Or at
Yep.
least according to the vendor, because they tested on all ethnic Han Chinese people in China, and it was all great, right? So then they sent it to Germany, where you have Turkish people, Norwegians, you have people from all over the world, right?
Yes.
Then they had a high level of false positives, but they're actually not, they're not really false positives, because they don't actually produce the same line as an antibody antigen reaction produces. So our machine can see the difference between a false positive based on insufficient detergent and a false positive caused by yeah,
Interesting. So you're
yeah,
saying that your machine might be able to help lateral flow assays increase
yeah,
their accuracy because
yes,
the false positive caused by contaminants or differences between chemistry of people,
yeah.
um,
And
you
you know
can actually
why?
distinguish that. Why?
Because the color of the line is different.
The color of the line is different.
Right.
Okay, so
A false positive from this method, or from this type of issue, is different. But there's no way you can train people all over the world to say, okay,
no,
if it's this shade of red, it's false positive. If it's this other shade of red, then this is another fault. You couldn't possibly do it, right? But
no.
a
But
computer
with the machine, you
camera,
can do it.
yeah, a computer camera with visual processing algorithm and light settings can tell that difference if you train the machine that way.
Well, class, this is
Yeah.
also fascinating. I feel like I learned a lot today. Best of luck with you on your machine. Please give me a heads up before. if once you guys really do break through the red tape, please give me a heads up before so I
Okay.
can be the first with the news.
Okay.
And what do you have? I know it's not your expertise, but I'm asking everybody this.
Yeah.
What are your predictions on what's going to happen in Taiwan? How soon will we be under control? Will we go to level 4? And when will we all be out in a brass monkey having a beer again?
Well, it's very easy. You can look at experience from the rest of the world, right? So in every other country who experienced this thing, in the very beginning, you had an exponential growth. You went from 4 people to 16 to 60 to 200, right?
Yep.
So there's only 2 options. It's either exponential growth or non-exponential growth.
Yep.
In every other country, it was exponential, 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 exponential. Right? In Taiwan, it was some numbers and some numbers, but now it's stagnated at 100 to 200 people. That's not exponential growth.
No.
So that means that they really contained the issue. So now they're just, Taiwan is in a great place, because I think what differentiates Taiwan from a lot of other countries, in my opinion, is people in Taiwan, they trust their government. I think that's a key differentiator between countries that have, success stories in here. It's because in Taiwan, you trust your government, and the government
do as you're told. Yes.
Right, they trust your government, and the government is transparent with the people, right? So it goes both ways, right? So that's just so I think now people follow. They have been good with things have shut down. People are careful. If you tell people here that to wear masks in public transportations or whatever outside, it's not a political issue.
No,
right?
No,
They don't try to politicize it, or it's not about your freedom. It's about trying to get a handle on this, right? So so my prediction for Taiwan is that we will reduce the lockdowns pretty soon. But when we do, right, or when Taiwan eases the restrictions again, we need to continue to be careful. Like Taiwan did so great for so long, right? And something happened. Something slipped through the cracks. We just need to get back to level 2 again, and then level 1, right? Need to go back, and then we need to put all these measures in place so it doesn't happen again.
Yep.
We need to test people when they come in. not this, not this 2-week quarantine without testing. Look at all these false. Okay, there's another thing I really should mention about PCR, because I've a lot of these interviews I've been doing because I have some PCR experience, right? Is PCR is great, is absolutely great because it's so specific. PCR is the only way you can really tell, other than sequencing, which strain of the virus is it that you're dealing with. Is the Brazilian, the British, the Danish, the Californian strain? Like, you can tell by using PCR exactly which strain it is. You can develop PCRs for that, right? That's great, but that's also a problem. So in countries like France, even in the United States, the FDA has taken some PCR kits off of the market because they're not able to detect the new strains.
Mm-hmm.
Because they're so specific.
They're too specific.
They're too specific. So if ask the virus, now if you see the mutation tree, right, it goes like, now it's like more than 200 different strains of mutants already.
Oh dear. So
Yeah.
PCR tests, while they are very good at telling you exactly which strain,
Right.
if it is a new strain that's not on the menu,
Right.
that might go completely undetected.
Yes, because you imagine somebody who, a company who early, who had a PCR, who wanted to sell a PCR test, developed a PCR test, several months ago, or maybe half a year ago, because they developed that PCR test based on whatever the biology was at that time, and they went through this whole red tape with the FDA and got approval from that test kit, and then finally they got to start selling their product, right?
Mm-hmm.
But now, the genomics have shifted. Now there's a different strain, right? So you can't possibly catch up with all the strains for using PCR.
Wow.
Because there's new strains coming out
really
all the time. So while everybody's screaming, oh, it's not approved, it's not FDA approved, and some of these FDA approved PCRs are actually not catching the new variants. The new variants. So that's the problem with PCR.
It sounds to me, I'm no epidemiologist, but it sounds to me like lateral flow assays and PCRs work great in conjunction, because you need a lateral flow assay that's broad and it's going to catch up,
Right.
all these like, it doesn't matter what strain, as long as you've got the COVID,
Yeah.
it's going to show up.
Yeah.
But then you will do a PCR, there's COVID there, and then you use the PCR to try and figure out which strain. And if you can't figure it out, then you might have a new strain.
Right. And that's what they're doing in Europe already, because Europe has said, we haven't had the problem here, right? Mm-hmm. So in Europe, they've had this problem now for a year, so they've figured it out. So what they recommend now is, if you have symptoms of COVID and you do a PCR that is negative, they recommend you to use a lateral flow assay to make sure we're not dealing with a new variant that doesn't have a PCR yet.
How many variants are there now? I feel like I've heard so many. I do believe the British variant is what's on the island right now, but I've heard that the Indian variant that's coming could be even more dangerous.
Right.
So
right.
It looks like we're going to take care of this wave, but it's not going to stop for a while until everybody gets vaccinated. And even then, I've been hearing news that the Indian variant could be deadly, even for vaccinated people. Right now, there just seems to be single cases.
Right.
But it does seem like something to keep an eye on. This is a wily foe.
Yeah. I did a podcast. I was guesting in a podcast where I talk about vaccines and variants as well.
Oh, great!
Recently.
Please send me the link. I think that'd be really great.
But the thing is, PCR is playing cat and mouse with the variants, obviously, right?
Well,
that's just how it is. And the same thing with the vaccines. So the vaccines are also
I totally get you can keep going on about this, and it's all very interesting. My brain is full, and I need to go and write my post now. But please do send me the podcast that you did on the vaccine, and I'll try and listen to it sometime today, okay? And I'll let you know when the post on LinkedIn is up.
Okay, sounds good.
Great. Thank you so much. It was great catching up.
Okay.
All right.
Bye.
Bye.
Bye-bye.